Hello students, I am Dr. Vinod Shinde. In the previous lectures we have discussed about the fermentation and alcoholic fermentation processes in which glycolysis turns out to produce alcohol, and in this case this lactic acid, in this lecture we will discuss about the lactic acid fermentation. In the lactic acid fermentation, it is a one-step reaction. Similar to the glycolysis in the mammalian cell, pyruvic acid is reduced to lactic acid, the main reaction being catalyzed by pyruvate reductase. Lactic acid fermentation is the characteristics of lactic acid bacteria, that is Lactobacillus family, in which these organisms causes spoilage of food. Although morphologically heterogeneous, they are characteristically producer of the lactic acid as the end product. The lactic acid bacteria are divided into two genera, that is two groups, that is homofermentative lactic acid bacteria and heterofermentative lactic acid bacteria. In the homofermentative and heterofermentative lactic acid bacteria, the homofermentative lactic acid bacteria are the member of genera that is Streptococcus, Leuconostoc and Lactobacillus. It is also found in animals also. In this type of fermentation, the predominant product is lactic acid, that is the homofermentative single state fermentation or single fermentation of lactic acid occurs, and predominantly we get a product that is lactic acid. The other products are formed only in traces or small quantities. Glucose yields two molecules of pyruvate through EMP pathway. This pyruvate is reduced to lactic acid by enzyme lactate dehydrogenase, that or pyruvate reductase. NADH2 is the hydrogen donor and is oxidized to NAD plus, giving rise to yield of 2 ATP molecules of the when the hexose is metabolized. Glucose through EMP pathway produces pyruvate, which then gives NADH2 gives away the hydrogen molecules to give NAD free radical, and pyruvate is converted into lactic acid. In addition to glucose, homolectic organisms also ferment other monosaccharides, that is fructose, mannose and galactose, and disaccharides like lactose, maltose and sucrose. These sugars are apparently converted into intermediate of EMP pathway and then to lactic acid. The final product in the lactic acid fermentation in the homofermentative pathway is lactic acid majorly. In the heterofermentative fermentation, this type of fermentation is found in some strains of Leuconostoc, Lactobacillus and anaerobic organisms, that is Eubacterium, Rhamnobacterium, Bifidobacterium, and in this fermentation along with lactic acid, ethanol, glycerol, acetate, propionic acid and carbon dioxide along with butyric acid are also found in addition to lactic acid. The major product in these cases is not lactic acid. In the heterofermentative pathway, we have number of products that occur that are produced from the single glucose molecule. Among the heterofermenters are included organisms that produce less than 1.8 mole of lactic acid per molecule of glucose. In addition to these compound, that is ethanol, glycerol, acetate, propionic acid, carbon dioxide and butyric acid are also produced in the lactic acid fermentation. Glucose is converted into pyruvic acid is then converted to acetaldehyde with the release of carbon dioxide and ethanol, then to ethanol. From NADH2 to NADH conversion, that is two hydrogen molecules are utilized here, and the free NADH is taken up by EMP pathway. Now pyruvic acid is converted into acetaldehyde and lactic acid in the lactic acid fermentation. In leuconostoc type of heterofermentation, acetate, ethanol and glycerol are formed in addition to lactic acid. In Lactobacillus brevis and leuconostoc species, EMP, HMP and ED pathway are absent, and these organisms possess a phosphoketolase pathway which in the overall reaction produces glucose to lactic acid and ethanol and carbon dioxide. Glucose is first metabolized to pyruvic acid, acetic acid and carbon dioxide. Pyruvic acid is then reduced to lactic acid and acetic acid is then reduced to ethanol through acetaldehyde formation. And glucose to pyruvic acid, then to lactic acid, glucose to acetic acid and then to acetaldehyde and ethanol. For every molecule of glucose fermented, there is a net yield of 1 molecule of ATP, 1 mole of ATP, and thus heterolactic fermentation produces only half of the energy produced during homolactic fermentation. Heterolactic fermentation produces only half of the energy of the homolactic fermentation. Here glucose is converted into glucose 6-phosphate, glucose 6-phosphate to xylulose 5-phosphate, which is then converted to glyceraldehyde, to pyruvate and to lactic acid. And glyceraldehyde, xylulose 5-phosphate splits into glyceraldehyde 3-phosphate and acetyl phosphate. Acetyl phosphate is converted into acetaldehyde and then to ethanol. And the glyceraldehyde 3-phosphate gets converted into pyruvate and then to lactic acid. In the heterolactic fermentation, we have number of products, that is ethanol, acetaldehyde, lactic acid, acetic acid. All these products are produced in the heterolactic fermentation of lactic acid through lactic acid bacteria. So homolactic fermentation and heterolactic fermentation are these two types of fermentation in the lactic acid bacteria, in which homolactic fermentation yields two times or heterolactic fermentation yields half of the energy of the homolactic fermentation, while heterolactic fermentation yields 2 times the energy of the heterolactic fermentation. So this is the lactic acid fermentation. We will discuss the next fermentation, that is mixed acid fermentation, in the next lecture.